Hello everyone and welcome back in our last day of training program, which is about the RNA pull down procedure to identify RNA targets of a long non-coding RNA. So just a quick reminder for the first, second, third day of our training program. So basically we cover the topics RNA pull down assay, and we explain what does it mean and on what it depends, and we give an introduction or background about the non-coding RNA, and then we explain in details the hybridization step, and in the third day we also show how to design the antisense DNA oligonucleotide probe that's specific for the long non-coding RNA of interest. And also we explained that the RNA pull down assay fall in 2 days. The first day start with a cross-linking step, whether it was culture cell cross-linking or tissue cross-linking, and how we fix the cells with the 1% paraformaldehyde solution, and how then stop the action of the paraformaldehyde, and then followed that with the rinsing steps, and then we collect the pellets with a scraper, and then we can store those pellets in -80, and if we want to proceed in the work, we can do the lysis step using the lysis buffer, and that contains the RNase inhibitor solution and protease inhibitors, and then next we did the sonication, and we explained how to optimize the sonication conditions, and then how we start sonication our sample, the lyse sample, and how to add the magnetic beads. And today we're going to explain the second day of the pull down assay, RNA pull down assay, the last step. So basically the RNA pull down assay, which is the second part or the second day of this assay, will be about RNA isolation step. So basically we have to follow these steps, 8 steps, in order to perform the second day of our RNA isolation step. So first we use magnetic support to separate the beads from the cell lysate. Then we discard the supernatant and wash the beads with a 900 microliter of washing buffer. And then we repeat the washing step 5 times with 5 minutes agitation on the rotator at room temperature. Second, we after the last wash, decant one last time and add 95 microliter of protease K buffer that contain tris HCl at pH 700 millimolar NaCl, 1 millimolar EDTA, 0.5% SDS, and add 5 microliter of protease K, 20 milligram per mole to the samples. And then after that we on ice, we work on ice. We thaw the input samples at 20 microliter, okay, and add 75 microliter of protease K buffer and 5 microliter of proteinase K. And then we incubate all the samples with proteinase K for 45 minutes at 50 Celsius, and then 10 minutes at 95 Celsius. And then we chill the samples on ice for 3 minutes before separating the beads from the RNAs with the magnetic support. We keep the supernatant and discard the beads. To purify the RNAs with RNA purification kit, which include a DNA digestion step, and then we store our RNA samples at -80 Celsius. Then we perform reverse transcriptase qPCR, real-time qPCR using RT kit, followed by qPCR using specific primers. Then we construct 2 DNA libraries corresponding to the 2 RNA pools obtained with each of the specific probes. Like for example, they use NEAT probe. Then we perform the sequencing on a next generation sequencing system. So basically we're going to show all these steps in picture to get better idea. So after we finish the hybridization step, after incubating our samples overnight under moderate agitation on the tube rotator at room temperature, on the next day we have to carry out the RNA isolation step and use magnetic support, okay, to separate the beads from the cell lysate. Then we discard the supernatant. So here they are just sucking the supernatant to discard it. And then we're going to wash the beads with 900 microliter of washing buffer that contain SDS 0.5% 2 times. And then we repeat that washing step 5 times with 5-minute agitation on the rotator at room temperature, and this is the rotator. And after that, after the last wash, decant one last time and add 95 microliter of protease K buffer, and that contains 10 millimolar tris HCl, pH 7, 100 millimolar NaCl, 1 millimolar EDTA, 0.5% SDS, and we add also 5 microliter of proteinase K. The concentration is 20 milligram per mole to the samples. So we have this already prepared proteinase K. We're going to add 95 microliter of it to our sample. So then we work on ice. So on ice we're going to throw the input samples that has a 20 microliter and add 75 microliter of proteinase K buffer, and then we add 5 microliter of proteinase K. And this is showing how we are adding these inhibitors. And then after that we're going to incubate all the samples with proteinase K for 45 minutes at 50 degrees Celsius, then 10 minutes at 95 degrees Celsius. Okay, now this is the 50, and then we change it to 95. Then we're going to chill the samples on ice for 3 minutes before separating the beads from the RNAs with a magnetic support. We keep the supernatant and we discard all the beads. Okay, so we work on ice, and then we don't finally get our RNA. So in this picture we are showing how we to suck or discard the beads and keeping the supernatant. And then now we have the RNAs, so we need to purify it. So we purify the RNAs with the RNA purification kit, which should include a DNA digestion step to eliminate the contamination. Then we start our RNAs at minus 80 Celsius for future use. So basically in the future we can do or perform the reverse transcription qPCR using ART kit followed by qPCR using specific primers, and then we can also construct 2 DNA libraries corresponding to the 2 RNA pools obtained with each of the specific probes. Then we can do or perform sequencing on next generation sequencing system. And by reaching this step, we reached the end of our training program, and I hope all of you get benefits or an idea or background about how we do the probing design, cross-linking, culture cell cross-linking, tissue cross-linking, how to do cell lysates or cell lysis or tissue lysis, how we do the sonication, how we do the RNA pull down assay, and thank you. And if you have a question, you can leave a question in the comments box. Thank you.